Okay, good afternoon. Thanks for inviting me for the opportunity to give the talk. I did say in the abstract I was going to talk mainly about counting rearrangement evolutions, but I thought I'd split the talk into two parts. So for the first part, I'm going to talk about the space of rearrangements that we get. So we've seen some talks of trying to get from one copy number confirmation to another, for example. But we've also seen some discussion on population dynamics. So I'm going to explore a little bit the idea that physics has been looking at ensembles of particles for a long time and developed various methods. So I'm going to explore whether there's things in there that we can use for ensembles of particles in biology. And by ensembles of particles, I mean cells, and see whether that's a good idea or not, as the case may be. So Vinny gave a talk that taught us how to explore the space of break and fusion bridges, in particular the inference, and deciding whether some structure occurs from break and fusion bridges. So there's an example of an amplicon. We've got some foldbacks that are going on here. So these are all signs of what we get when we have a break and fusion bridge process. And we've got copy number increases. And then we've got this kind of folded structure going on. So the DNA is wrapping all over the place. And this is kind of a signature of what we get from a break and fusion bridge process. So just a quick recap on what goes on with a break and fusion bridge. So without getting into the biology, we can think of it as a break is occurring at some point. We get duplication up to that break. So we get a kind of symmetric situation going on here. So we've got two symmetric things. And in some sense, those two things get glued together. And that process continues, and we end up with these folding structures. So the question I'm going to consider, and I'm not just considering it for break and fusion bridge processes, I'm thinking of there's different mechanisms that can happen. There's break and fusion bridge. There's tandem duplication processes we've heard about. There's inversions as well. So there's different things that can affect the way that these amplicons are structured. And so when we're kind of trying to get from one genome to another, it's of interest to kind of say how big is this space that we're exploring, and that's kind of what I'm going to talk about for the first half. So given k events, so there could be k break and fusion bridges, there could be a mixture of events going on, we can ask how many evolutions are possible. So that's kind of what we mean by saying how big is this space. And so what we're counting is these evolutions. So what I mean by an evolution is a sequence of genomic configurations. So we're starting from a wild type, and then every time we've got one rearrangement, we've got a new structure. And what I mean by an evolution is that entire set, that entire sequence, I should say, of genomic configurations. So we're kind of asking how many are there. So for break and fusion bridges, I'm just going to talk you through how this space kind of grows. So there's just, we can think of that line as a reference, and this is one breakage fusion bridge, so you get one fold that goes on. So if you've got one break and fusion bridge, it's not a very big space, you've got one thing that happens. But if we allow a second fold, we can break at the top or at the bottom, and that gives us two possible structures that can arise. So if we allow two breakage fusion bridges, the space is of size 2. So then we can say what happens if we have a third event. So again, we can break it in two places, and that gives us two possible structures. The alternative is that we try and do more breakage fusion bridges on that structure. So if we do that, we can either have a third break in one of these two positions. So this gives us two more structures. Or we can have a break in this position here, which gives us 4 possibilities that goes on. So we get 4 structures that come out. So we can kind of build up a picture of what this space is like as we keep doing these break and fusion bridge operations. And when we do it, we get this kind of, so here's a count of the break and fusion bridges we've got, and here is a count of the number of evolutions that is possible. So it's kind of growing in this slightly unusual power of 2. And so we can write down a formula for that and get some idea of the size of the space. So that's if we restrict ourselves just to break and fusion bridges. We can think about doing similar things with tandem duplications. So again, I'm going to align everything up to a reference here. So this is the reference, the wild type, if we like. And so we start with the normal genome, and then if we've got a tandem duplication, we've got some bit of DNA that we want to copy. So the green bit we're going to copy, so we've got a green bit of DNA there. And then we're copying the piece of DNA and shoving it next to itself, so the ABC becomes a BBC, if you like, algebraically. But we're lining these up to the reference, so these two bits are both lined up to the same place, and we've got a connection from one end to the other. So this representation is a kind of visual representation of what's going on with a tandem duplication. So we can keep going. So for example, the red bit there could be the next bit that we duplicate. So we've got one copy of the red bit, then another copy of the red bit. So this would be the next structure we'd have if we allowed two tandem duplications to take place. And we can keep going. We can copy the blue and the purple. And you can build up pictures of what kind of sequences of tandem duplications look like if we allow them to continue. So we can ask similar questions, and if we have tandem duplications going on, so how many structures can we get? And I'll not talk through this in too much detail, but these are all the configurations we get if we allow ourselves two tandem duplications to occur. So we get 11 structures in total. So when we kind of explore this, if we allow ourselves one tandem duplication, we get one configuration. If we have two, then we've got 11 configurations, which are all these things here. And we can build up this rapidly growing sequence, which is a very big space. And there is a formula for this. So it's a product, it's similar to the broken fusion bridges in that we've got this power of 2 in some sense that dominates. So what we'd like to be able to do is to mix these things up and not just restrict ourselves to broken fusion bridges. We want to allow lots of different mechanisms to take place and then ask how big is the space of rearrangements that we can have. So how can we go about this? Well, here's a reference. So let's allow ourselves one tandem duplication to take place. So we've copied this bit in the middle, so that bit's duplicated twice, and that gives us one structure. And so we allow ourselves a second one, and this is a breakage fusion bridge event, so that gives us a palindromic bit of DNA. So if we imagine we break it here, so that's the original structure, and then copy it backwards. This is what we get if we have a break-in fusion bridge following a tandem duplication. So if we do that, we've now got 3 breakpoints that have arisen. So when we're first exploring this space, it's quite natural to try and add more and more operations below and then try and come up with some way of working out how big the space is. But it doesn't work terribly well. What you actually have to do is kind of go backwards in time rather than forwards in time. And so I've got this reference here, and we kind of assume that the reference is this gold standard that's not been touched. But what we can actually think is, well, let's suppose that this reference is actually a product of a preceding operation. So there was a broken fusion bridge, for example, that happened before, and this reference is actually the product of a broken fusion bridge. So when we do that, that's no longer the reference. We go back in time, and there's actually an earlier reference that's the true reference in some sense. And we've now got this sequence of 3 breakpoints that kind of run along the structure in that order. So although this is now folded, we do have 1A, then 1B, and 2. So that order is kind of conserved. So then what we can do, we've kind of got a bit of flexibility when we're exploring this space. As long as that order is conserved, we're okay. So we can move these things around and see what different configurations we get. So that's a representation of those 3 breakpoints that have been put on that structure. And actually, when we think about all the different positions that we could place those 3 breakpoints, we get 2 to the power of 3 different structures. So there's 8 structures there. I say 2 to the power of 3 because the 3 is actually the number of breakpoints that occurred afterwards. We had 2 from the tandem duplication, 1 from the broken fusion bridge. And for each of these choices, we're going to get different copy number configurations, so the space is going to be different. So by kind of going backwards in time and resetting the reference, we can actually say how many different configurations we're going to get. So we just saw that choice. That was the break confusion bridge. So we had 2 to the k choices in general, where k is the number of breakpoints. So that's the number of choices where we did that one. The Tandem duplications, I've not talked about where this form comes from, but you can do quite a similar argument as to this one to say where this factor comes from. And so this gives us 4 possible choices. So for this formula, we're substituting in 1 because there's 1 breakpoint that comes afterwards. And then finally, we've got the last one. And this has no follow-on event, so we only actually have 1 choice here. So we can kind of put all this together, and we just multiply those choices together, and it gives us the total possibilities for the space. So we're restricted in the sense, I'm now saying, let's have a break confusion bridge, then a tandem duplication, then a break confusion bridge. And I'm asking how many different structures can you get if you do them in that order? And then the answer is that it's 32 different possible configurations arise. So we can do that in general for quite a broad sequence of different types of rearrangements. We fix the things that we want, and then we just multiply them together based on this induction that's kind of going backwards in time rather than forwards in time. It's resetting the reference each go. Yeah, so we can do it in general. So I give you the terms for those two, but we can do it for inversions. We can do it for inverted duplications. And by redefining the reference, we can say how big the space is. I mean, one issue that's come up a little bit is that I assume unique breakpoint use in all this. So I'm assuming that every time we have a new rearrangement, it's hitting the genome in new places. So, I mean, that's open to debate whether you'd want to assume that. But if you do assume that, then you can do this counting mechanism to say how big the space is. And as a rough guide, for when you put these operations together, the size of the space is roughly like 2 to the power of n squared. So the size of these spaces of genomic configurations is growing faster than exponential. It's a big space to explore. So that kind of means that when you're looking at lots and lots of rearrangements and you're trying to explore this space, it's going to become quite difficult quite rapidly.
Chris, just a quick question. Obviously, this is the number of unique outcomes with distinct order of events. You can get the same sequence with a different set of events, different order of events.
Almost. So you can have the same starting event and the same last event, actually. If you've got genomic configurations that are different in the middle and you end up with the same structure, then according to this definition, the evolutions are different, even though the structure you've got is the same again.
The number of unique evolutionary paths.
Yes, exactly. Yeah. Okay, so that was a bit about rearrangement. So it was a nice little puzzle that we can solve. The second half is somewhat more speculative, I must admit. So the system I've got in mind is the following. So we can think of this as almost as a pre-cancer system rather than a cancer system. So the thing about physics is quite often these systems are quite nicely controlled, and biology is a bit more chaotic, especially cancer systems. But one of the nicest controlled systems is actually skin tissue, because you've got this single layer of tissue, and there's only 2 cells that actually exist in this layer. You've got the proliferating cells. Now they can divide and produce other proliferating cells, or they can produce differentiated cells, which rise up and form the top layer of your skin. So in terms of the dynamics, the only thing we really need to worry about is this basal layer, as it's called. There's quite nice, precise experiments that biologists can do. We can watch cells, we can see how they divide. This is an example here of a staining experiment where one cell was stained and then this single layer was observed and the staining spreads out. So you can kind of see how this clone, in a sense, is spreading out. I mean, just a point to note, if you stain, say, 100 cells in this tissue, then what you tend to find is that most of those stains actually disappear, and you end up with one very big clone that kind of dominates the whole area. So that does have implications in terms of mutation accumulation, because although this is a clone, we can think of these 100 things as a mutation that arises. And so for a skin system, what we would find is that most of the mutations that arise in the skin tissue itself will just drift out of the system. And it's a very, very small subset of cells that will actually end up dominating the layer. So just by drift, we're going to find that mutations accumulate and kind of sweep over the tissue. And there's no selection that would go on there. So the idea of this, and I'm not going to get this far, is that we have cells. We've got nice information about cells in terms of how long it takes a cell to divide, what the mutation rates are for an individual cell. I mean, that's actually quite hard to get, I think. And then selection we can think of as acting on the cell in terms of whether the speed of division speeds up, for example. And then in terms of the population, we're interested in things like the size of the clones that we're getting, the tree structures that we've heard people talking about, and we've seen from SORAB some of these mutation sweeps that occur. So these are kind of macroscopic things, and these are microscopic things. So in terms of population dynamics, it would be nice to be able to join these two things together. So that's the holy grail. So in terms of population dynamics, the temptation is to use Markovian dynamics. They're very popular, and I think they're popular because they're convenient, because the maths is quite nice. So in terms of cells, I'm just drawing attention to some limitations. If you assume Markovian dynamics, you assume that the cell cycle is actually exponentially distributed. Now this is data from cells, so cells have been watched and they've been timed to see how long they take to divide. And you do that for lots of cells, and you build up a picture, and this is what their life cycle looks like. So it's reasonably close to a gamma distribution, and it's nothing like an exponential distribution. So you can then say, so what? Does that really matter? Well, I think it does in terms of the way that these things grow. So this is just a simple cell population growth. If you assume it's exponentially distributed, for 1 unit of time, you get this explosion of e to the power of t. At the other end of a Galton-Watson type model, where you've got one cell, then it divides at exactly 1 time unit, you get 2 cells, then 4, then 8, etc. Then the growth is different between the two. And actually the real data, the gamma distribution, sits somewhere between the two. So the growth is going to be a lot slower when we're dealing with gamma distributions than it will be if we assume Markovian dynamics. So the lifetime distribution, in terms of modelling the way populations change, is quite important. So in terms of ecological modelling, this is often referred to as age structure. So the age structure is quite important. Another aspect that can occur, so this is just a simulation. So we take one cell, we'll let it divide, or we'll let the population have 99 cell divisions. So we're going to end up with 100 cells. And then if we assume we get of the order of 1 mutation per division, we can play around with that parameter, we can then ask how many cells have lots of mutations and how many cells don't have many mutations. Now this is the standard Markovian model, and what we find is we can find lots of cells that don't have many mutations, and we can find cells that have plenty of mutations. Now, in terms of kind of transitioning, if you're trying to accumulate mutations, then this is going to be an easier system to accumulate, but this is the one that's a bit more realistic. So in reality, it probably takes clones longer to pick up mutations than standard Markovian dynamics would do. So I'm not going to get into the specifics too much. I'm just going to draw analogies. So in terms of physics, we've maybe seen Feynman diagrams before, where you've got 2 particles come in and then 2 particles come out. The particles change type. Now this is what we might be a bit more familiar with. We've got a normal cell. It changes into a mutant, so it's changing species, just like we have in physics. And we get death as well. So the other thing is that we've got particles changing in number. We've got cells changing in number. So there's a lot of analogy that goes on here. And I mean, it was noticed in the 70s and 80s by these two guys. I mean, there's a guy called Dye who worked out how to use this kind of methods for chemical reactions. And Politi in the 80s worked out how to use this kind of machinery from physics to actually have a go at birth-death processes. So there's a question of whether we can kind of do the non-Markovian version of what was going on with Politi. So I'm just going to kind of go through the two different fields, if you like. So what we can think of is that quite often in physics, we've got Q is the position of a particle, and I'm going to say in biology, we've got A is the age of a cell. That's the analogy. So in physics, we've got states. So I'm going to say that this, so where that's a state of 3 particles, I'm going to say this is a state of 3 cells, where those are the ages of the 3 cells. In physics, we have a superposition, which we can think of as a wave packet. This is a superposition, but this has a real probabilistic interpretation because this function here represents the probability of having n cells with a certain spread of ages. But perhaps most importantly is the following. So here we've got the evolution of states equation that we get from Heisenberg. And what we can do is we can take the way that birth-death processes evolve, and we can write out an equation that is very close to what you get in quantum mechanics. So H is the Hamiltonian, and this thing here is an operator that describes how the birth and death goes on. So it's not just an analogy like we've got at the top of the board here. The fact that this is true as well means we can really treat this as a dynamical system and use the methods of quantum field theory. So I'm not going into the details, but for example, here's a kind of matrix element. This gives you the probability density function. Another thing that population dynamics are interested in is the distribution of the size. We can also get that out. Feynman diagrams are quite famous, but in a very real sense, if you actually go through the machinery, the Feynman diagram that you get here, when you turn it into the biological problem, this thing here that we think of as a tree representing a birth-death process is actually the Feynman diagram for this system. And so what you can do is you can use all this machinery and you can actually construct path integral representations of all these different systems that goes on. So it gives you a completely different set of machinery that you can start to think about these problems. So we've done that. So I'll just finish. I mean, this was a curve that I had earlier. So there we had the exponential growth of the Markovian system. This was the Galton-Watson process where it's stepping up. And the blue is from the real data. So you can kind of use all this machinery and you can come up with expressions describing the blue is the growth of the population there. For gamma distributed systems. So you can use this machinery to start to get information out about how populations are growing. So having said all that, I mean, the caveat is the following. I mean, the aim of this, which I've not got as far as doing, is to try and analyze skin systems to describe how clones are growing and how the mutations are spreading through something, where we've got a system that is quite nice to control. So I've not got that far. All I've actually done is model a basic system. So there's the possibility that we can use these physical techniques to actually analyze some of the systems that are a bit more well-behaved. Whether these methods from physics will translate to cancer systems where the behavior of the cells is a lot more chaotic, I don't know, but it would be interesting to try. Yeah, so I mean, the other thing to add is that quantum field theory, although it looks at small particles, I mean, there's a lot of quantum field theory that does material physics. So material physics has lots and lots of particles, and similarly with biology, we have lots of particles here. So, and given that there's been decades of people doing this stuff, there is the possibility that there might be methods there that we could use to try and bring together information about cells at the microscopic to say what's going at the macroscopic level. finish, I'd just like to thank Tom Chow. We did the work on the aging structures. Luca and Tao Yang was the rearrangements, and then I got data from Peter Campbell, and these guys were responsible for doing the cell watching. But I'll stop there and take any questions.
Questions? Let me start. A natural question about the first part. The more common situation in the literature is where you have a target permutation or genome, and suppose you know how to compute the shortest path. Can you tell how many alternative solutions? Yes, are your techniques able to make a dent at this?
The short answer to that is no, I think. I mean, it's an interesting question. So the business of counting the evolutions you can do because the structure is allowing you to do that. But if you say something like, here is this copy number configuration, how many different paths are there to get to that configuration? I don't have good answers to that question, but it would be nice to see.
Can you even calculate the shortest path?
Well, yeah, I don't know actually. I mean, for inversions, we know we can, but and for break and fusion bridges, I guess you
yeah, just the FP maybe, but if you mix it up.
Yeah, I don't know. Yeah.
If you intersperse deletions into one of the events that could happen, that actually constrains the space because if you lose an allele, then about mixing in deletions into that.
I haven't. You do see that happen with break and fusion bridges, for example. So you can have a sequence of break and fusion bridges where the break and fusion bridge deletes folds from earlier break and fusion bridges. So you see you've got 5 folds in the system, but you've no idea whether it was 5 cycles or whether it was 50 cycles, and they kept, deleting stuff. So I have been ignoring that. But you're right, it will constrain the system because you'd either get down to one allele or the DNA is gone.
Okay, thanks again.